Now, activation requires binding of the antigen. In case of our B cells, there is the receptor called B cell receptor, which is BCR, and that binds with native antigen. It doesn't require any kind of other receptors along with the antigen. So B cell has its own receptor. T cells will have TCR, which is T cell receptor. It requires antigen binding with other receptors. So it will be processed antigen, and you will require MHC molecules for activation of T cells, right? And further, what will happen, and they will undergo mitosis, and also they will undergo clonal expansion. So you get different clones of these cells. So the cells will be targeted against those antigens, and those kind of similar cells will be there. So that's the clonal expansion of the cell. Now, one by one, let's talk about these lymphocytes. Let's talk about B lymphocytes. They are also known as B cells, right? So let me write it down, B cell over here. The maturation and development, if we talk about maturation and development, will happen bone marrow, and then further development happens because of the VDJ recombination, and that contributes for the receptor diversity in B cells. Okay, that's a big topic. Now, if we talk about the receptors that are present on B cells, let me design the cell, and let's design these receptors one by one. The first and far most important receptor in B cell is the BCR, B cell receptor, right? And you know that it's the IgM or IgD antibody that is, IgM and IgD antibody that can bind directly to the native antigen. And then further, you have CD79A. It's also known as Ig alpha, and then you have CD79B also. Okay, B also, that is also known as Ig beta. These are really important receptor, and they transmit activation signals inside the cell. Next is CD19 receptor. You have CD21 receptor. They are co-receptors, and then you have CD81 complex. So CD19, CD21, and this is CD81 complex, and all these receptors, they are required for enhanced sensitivity to antigen. Next is MHC class 2 receptor, which is important for processing the antigen to the helper T cells.